All right, let's move on now to our next speaker, Jeffery Hansen from Colorado State University. And he's going to talk about, are chromosomes organized as globules of globules? Role of nucleosome interactions. Jeffery.
Thank you. I want to get started by going over some information that suggests that the age-old paradigm involving chromatin and how chromatin folds into a chromosome is changing. Let me start by making a couple of points about chromosome territories that you don't normally hear. One is that in order to form a territory, the interaction of the chromosomal chromatin fiber must be self-interacting. Okay, the chromatin fiber is interacting with chromatin fibers within the same chromosome, not with other chromosomes, except at the periphery for the most part. The other thing is that you rarely hear, but is quite clear from this picture, is that chromosome territories are globules. So the final endpoint, structural endpoint of an interphase chromosome is a globule. Now here's the dogma. You can take any of these models from textbooks, from reviews, and they all say the same thing, which is the DNA is packaged with histones, and that the next step in the formation of a chromosome is the formation of a 30 nanometer fiber, which is a kind of a helical structure. Most of these models skip the interphase chromosome altogether. In some cases where they put it in there, they show it as spaghetti. So this has dominated the thinking from the chromatin perspective, not so much from the chromosome perspective, but from the chromatin perspective. And I don't have time to go over all these different papers, but in the last several years, it's very clear that by many techniques that you cannot find 30 nanometer fibers in bulk in the nucleus. Okay, so this has recently been reviewed if you're interested in going into these papers in more detail. But microscopy, small angle X-ray scattering, high C data all say the same thing, that 30 nanometer fibers are not present in bulk in chromosomes. And what they do say is that let me just put that up there. The default state of the genome is the 10 nanometer fiber, okay, the extended form of the chromatin fiber. And moreover, this 10 nanometer fiber is packaged together in an interdigitated form, kind of like my fingers here coming together. Okay, so if it's not a 30 nanometer fiber and successive coiling, what is going on? And we've heard a great deal about high C. And let me just remind you that if you look at the very high resolution high C data, that you can conclude that eukaryotic genomes consist of A and B compartmental domains, which are defined by the chromatin signature and transcriptional state. And in vertebrates, you have these CTCF loops that demarcate the genome. And we've heard about those in great detail. What you don't get from high C data are structural information. What are the structure of these domains that you see here? What are the physical structure? So this is work from the Olens and Olens lab that was published a couple years ago. And this is immunofluorescence microscopy using an antibody that recognizes a domain on the surface of the nucleosome. And you see this punctate pattern staining where histones are in the nucleus. Okay, and each of these puncta correspond to a globule of approximately 200 nanometers in size. And so the model that they put forth, and they show this starting off as a loop, but the important point is that these loops are really a globule of very densely packed nucleosomes, 500 to 1,000 nucleosomes per each one of these puncta. Okay. Recently, super-resolution imaging microscopy has revealed the structure of specific loci in chromosomes. Here's a paper where they looked at active compartments null compartments and inactive compartments. They saw a difference in the compaction level between these different functional states, but in all cases, the individual loci that they're looking at are globular. You saw this yesterday in Era's talk. This is the modeling that they've done. And again, this segment of chromatin consists of a number of different globules that are strung together. And then the one that I like the best was recently published in Science. And they looked at a great number of different chromosomes, individual loci within 4,500 different chromosomes, and get somewhat different structures, specific structures, depending on which cell they looked at. But by and large, everything looks pretty similar here, and they're globular. Okay, they've color-coded the length of the segment of the chromosome they're looking at, so you can look at it. These domains are globular, separated usually by regions of extended chromosomal fibers. Okay, so we think there's evidence for a new model, which starts with the nucleosome. As I said, the default state of the genome is the 10 nanometer fiber, which exists as an irregular zigzag. This would be an example of packaging of 3 different segments of the chromatin fiber in an interdigitated polymer melt type structure. This packaging ultimately leads to formation of compartmental domains which are globular. And then you subsequently have AA domain interactions and BB domain interactions and a large number of steps that we don't understand to ultimately give you a globular chromosome territory. And I think that it's important to point out that at every step of the way here, starting with the nucleosome itself, which is a globule, you have globules which are forming higher order globules all the way up to a globular chromosome. Okay. So that's the model that we would put together based on in vivo data. What about in vitro data? Is there anything about chromatin that's consistent with this new way of looking at chromosome structure? And the system we use in my lab to ask this question has tandemly repeated nucleosome positioning DNA. You reconstitute this DNA with histones to get a chromatin fiber. Everything's done with either wild type or I'll show you very quickly at the end, mutant histone studies. The important thing here is that this gives you a length and compositionally defined fiber, 12 nucleosomes long. The length of the DNA is the same in all of the molecules. The positioning of the nucleosome is the same in all of the molecules. So it's a wonderful model system that allows you to deal with the complexity of chromatin. 12 nucleosomes on a 2.5 kilobases of DNA has a mass of 3 million Daltons, and I'll show you some evidence that this is the monomer in our system. 3 megadalton piece of chromatin is a monomer in our system when it self-associates. So if you take this system and you put it in the presence of salt in vitro, the first thing that happens is it folds into a 30 nanometer fiber, and that's the basis of why so many people believe that the 30 nanometer fiber was a chromosome assembly intermediate. It certainly happens in vitro, and my lab has studied this for the better part of 25 years. But something else happens if you go to slightly higher magnesium concentration. So this happens in 1 to 2 millimolar magnesium. If you go to 2 millimolar or greater, then these nucleosomes, these chromatin fibers self-associate, which are interfiber nucleosome-nucleosome interactions, and they form large aggregates that pellet very quickly in the microfuge. Okay, and this is what I mean by the model system being a monomer. This is the monomer. It self-associates. Okay, so the dogma when it comes to this phenomena in my field is that it's been an insoluble aggregate or a precipitant, and it's been ignored for almost as long as people have been studying chromatin. But I'll run through some data very quickly that show you that we believe these are biologically relevant structures. Published data. Okay, so the first thing is if you look with fluorescence microscopy at them, these are the kind of images that you get. And it's quite clear that these aggregates are globules with diameters ranging from 0.1 to 1 micron. I don't have time to show you the data, but we did analytical ultracentrifugation on these samples, and rather remarkably, they sediment between 5,000 and 300,000 S. You think about something that's large normally, like a ribosome, is 80 S. These are 300,000 S. They are the size of chromosomes themselves. Transmission electron microscopy, these are 3 representative images of what we see of fixed and negatively stained samples, again, is consistent with these structures being globules, okay? And the negative stain, any place that it's darker, this is a furrow in the, and so you see that it appears as if these globules are formed from smaller globules, okay? Globules of globules. If you have a globular structure and you know the sedimentation coefficient, you can calculate all kinds of interesting biochemical parameters. Really, the only one that the point I want to make right now is that these structures that form in vitro range in size from a fraction of a chromosome all the way up to an entire chromosome themselves. Now, if you go to high magnification with the transmission electron microscopy, where these arrow points are right here, you see individual nucleosomes, which are 10 nanometers in diameter. You see no 30 nanometer fibers. They're just packaged together. Again, I don't have time to show you the small angle X-ray scattering, but that's consistent with these images. There are no 30 nanometer fibers in these globules. They're made from 10 nanometer fibers that are packaged together. Okay. Recently, the Nordenskjöld lab published a paper where they looked at T4 phage DNA that had been reconstituted into nucleosomes. You don't get a very nice chromatin sample when you do that, but I think the results are still valid. And the results show that here's the DNA molecule alone. This is fluorescence microscopy again. Here is the T4 DNA reconstituted into nucleosomes in the presence of salt. It condenses into a globule. Okay, so there's nothing artifactual about the short arrays. They're doing the same thing and forming the same structures as a 166 kilobase T4 phage DNA molecule. Now, if you go in situ and you take isolated HeLa nuclei and expose them to low salt structures that dissociate these aggregates in vitro, okay, you put them into low salt, they fall apart. They require salt to be stable. If you do that with nuclei, rather remarkably, you see that the nuclei swell in size and you lose all internal architecture. And so, and the cohesin CTF linker histone content of these nuclei are completely unchanged. So the same solution conditions that dissociate the globules in vitro dissociate the nuclear architecture and disassemble the nuclear architecture in situ. Okay, so we think that the nucleosome-nucleosome interactions that stabilize chromatin globules in vitro are the same interactions that occur in situ, and that essentially the self-association of 2 short chromatin molecules mimics the long-range interactions of the chromatin fiber within a chromosome. So if we were to make a model of the in vitro results, we start with nucleosomes, 10 nanometer fibers, which interdigitate to form a polymer melt, which then form globules, which upon further self-association form higher order globules. It's the same model. Okay, so to answer my own question, yes, we believe that there is lots of in vitro data now in the last several years that's consistent with this new view of looking at the way that chromatin contributes to chromosome structure. So let me finish with unpublished data. I want to show you some FRAP, 2 different ways of doing FRAP, and 1 slide at the very end about core histone tail domains. Here's the FRAP of the whole globules. Here's the globule, and here is the bleached globule after 24 minutes. There's no exchange between the solution and the globule. Okay, it is not behaving as a liquid. And if you partially bleach the globule, you see the same thing. Here's a partial bleach of two different globules, and it doesn't recover. So there's no recovery within a globule. So these chromatin globules are solid. They're not liquid, and that may come as a surprise. But if you think about it, and we just heard these wonderful talks at this meeting so far about liquid condensates, I'll just pose the question, is it possible that chromatin provides a solid state support for liquid condensates so that everything in the nucleus is not a liquid? You have liquid condensates that are in the midst of a solid state substance, which is chromatin, which is present densely throughout the nucleus. If the answer to that question is yes, then that would make the nucleus a gel. Now, if you do FRAP with cyber green, which is a small DNA binding fluorescent dye, you get a different story. So, I have cyber green in our samples, and we FRAP the globules, and then we say, do they recover? And when you use cyber green, they do in fact recover, but they recover about half of what the original fluorescence was. And the percent recovery is very much salt dependent. So the packaged chromatin fibers essentially form two compartments. Okay? One is freely accessible to cyber green, and one is completely accessible, and partitioning between the two compartments is salt dependent. And of course, this is of interest because you've heard that the genome is partitioned into A and B compartments. And most of the attention is focused on the proteins that are involved in that process. We wonder at this point whether fundamentally chromatin isn't capable of doing that on its own. Okay? And what the other proteins are doing are shaping and molding and modifying what's going on. Fundamentally, the process is set up by chromatin. Okay, so what is responsible for the phenomena that I've been telling you about? Here are the histone octamers that the DNA wraps around to form the nucleosome, the crystal structure of the nucleosome. Something about these histone octamers are dictating everything I've been telling you. It's the only thing. The DNA doesn't do this on its own. It's the histone octamer. And let me just remind you that although you usually show the crystal structure of the histone octamer, that in fact the histone octamer has 8 intrinsically disordered N-terminal tails. And when you put them in there, even in this cartoon form, the nucleosome goes from being this beautiful structure to something that isn't quite so beautiful. Okay? And all these tails are on the outside out into solution. The other thing about the nucleosome that's fascinating is it has this patch on the surface, the acidic patch, where you can just see here, and it's a combinatorial interaction domain for many, many proteins as well as the histone tails themselves. So the last slide, the last data slide here, are what we've been doing recently with mutant chromatin fibers. And we've been making these model systems with histones that lack specific tails. This just shows you a differential centrifugation assay, essentially a pelleting assay. It's our first approach to understanding what's going on whenever we do something new before we get into the other techniques that I showed you. Okay, so here in red is the wild type arrays. If you start to take off the tails, here is the H4 tail removed, and it's shifted to the right. You need higher magnesium, but the phenomenon still occurs. And if you only have the H4 tail, it also still occurs, although again, you need higher salt concentrations. The acidic patch mutant is very interesting because you need less salt. And the most likely explanation for that is that when you mutate the patch, the tails cannot bind to that patch. The only place they can bind to is the DNA. And that means that there'll be more tails bound to the DNA in the patch mutant, and you need less salt to induce the phenomenon. So if you think about it, if salt can replace the function of the missing tails, as long as one is present, one tail is present, it implicates linker DNA charge neutralization in the function of what the tails are doing. So charge neutralization is clearly extremely important in this system. The H4 tail alone can mediate self-association. We found this quite remarkable. Only 25% of the total tails are there in the form of two H4 tails. So we wonder whether only subsets of the tails are involved in stabilizing these globules in the wild type, okay? And the acidic patch mutant, as I mentioned, requires less salt. It's involved, but it's not required, okay? So something very important is going on, but it's not absolutely required. So the take-home messages, if you can remember four of them, are that an array of 12 nucleosomes self-assembles into chromatin globules that range in size from compartmental domains identified by high C to entire chromosomes themselves. The chromatin within those globules partitions into two compartments, a compartment that's accessible to small molecules and a compartment that's not accessible to small molecules. And we really don't know at all at this point what's going on there. We're in the process of trying to figure that out. Histone tail DNA and tail acidic patch interactions are what mediates the chromatin fiber self-association. And I'll end this slide with the rather audacious statement that we think that the information needed to assemble a globular self-interacting chromosome territory fundamentally lies within the nucleosome itself. Okay? And again, I kind of mentioned this earlier. Other proteins that are present are important for shaping and molding the chromatin within the nucleus. But the chromatin itself seems to be able to form globules, and remember that's what the interphase chromosome is a globule, the size of those chromosomes in vitro. And if you can only remember one thing, this is what I would like you to remember from this talk. When you think about the structure and the functions of the 3D genome, don't forget the nucleosomes and their self-interactions. They're very, very important. Thank you.
Questions?
Okay, thank you very much. I want to ask you whether you rule out 30 nanometer fiber at all.
I didn't catch that last part, sorry.
Do rule out the existence of 30 nanometer fiber at all?
Right.
Because you are showing the results of human cells. What about the chicken aerocyte
Right.
cells?
My feeling is that the 30 nanometer fiber must exist. It's a reversible transition in vitro. It's been studied to death. I just don't think it's an assembly intermediate in chromosomes. Okay, so it's probably there for regulating gene expression in certain specific locations in the nucleus, but it doesn't seem to be necessary to form in order to form a chromosome.
Okay, so it is existing in interface nuclear, okay?
Yeah.
Phil?
Since these interactions are so electrostatic
Yes.
in forces, have you ever considered mixing in RNA and asking what RNA does to formation of these globules?
No, we have not done that. That would be a great experiment. It would be a great experiment. You know, we were very surprised several years ago when we started studying this phenomenon, and what I talked to you today is pretty much what we know. We believe that these are valid in vitro model systems for chromatin contributions to a chromosome, and we want to start adding proteins and as you said, RNA back to begin to more realistically simulate a real chromosomal environment. I think that would be a great experiment.
One more.
So in your model for chromatin globules,
Yes.
what do you envision different chromosomes actually forming one big genomic globule, or what really stops it from different chromosomes fusing into one globule?
Why don't chromosomes interact more, different
Yeah.
chromosomes with themselves? Yeah, I get that question quite frequently. And all I can think of is if you think about a mitotic chromosome, it's a self-interacting rod, right? And at the end of mitosis, when the nuclear envelope reforms around the chromosome partially decondenses into an interphase chromosome, but I think it's just simply remaining a self-interacting territory that it started off in a mitotic chromosome. So I don't think I don't see any reason why it couldn't interact with other chromosomes, but I don't think that it ever has the opportunity to, I guess is what I'm saying.
Okay, thank you very much.